Our last speaker for this session is one of our own graduate students, Shariq Mujib. Shariq is a PhD candidate with the IMS and presently in his year 5 of his program, which is in the doctoral stream program, working under the supervision of Dr. Mario Ostrovsky. Shariq got his undergraduate degree in biochemistry from the University of Waterloo. Presently, he is a recipient of multiple provincial and federal awards, such as the Queen Elizabeth II Graduate Scholarship for Science and Technology, as well as the National CIHR Doctoral Award. He will share with you some of his research work on HIV and AIDS immunology. One of the mechanisms for viral persistence involves HIV virus establishing latency, which means they're going into hiding from the immune system. What Shariq's aim is to improve the immune system's ability to detect and hence eliminate the virus by using small molecule inhibitors against an HIV protein critical for immune evasion. Shariq hopes to defend his PhD very soon, within the next year, hopefully. And please welcome Shariq to render this presentation.
Morning, everyone. I'd like to start off by thanking the IMS and the organizers for inviting me to share some of my data. I have a bit of a sore throat, so I hope it doesn't come out right now. So I'm a PhD candidate with the IMS, with Mario Ostrovsky, as Vasu mentioned. I'll be highlighting some of my work. It's basic science work, all on primary cells, immune cells. And what I try to do is try to eliminate HIV from latently infected cells. I wanted to start off with a quick primer on HIV infection. So what is HIV? It's the human immunodeficiency virus. It is transmitted via blood contact primarily, or other mucosal fluids, but usually it's by blood, and unprotected sex is one of the main modes of transmission. Primary target of cell of infection is a CD4 T cell, which is an immune cell critical for control of any infection. And so what HIV does is it kills these cells in time. And so once you lose these cells, you become immune compromised and become susceptible to random or opportunistic infections, which eventually lead to death. HIV is the virus that causes AIDS, as you guys are familiar, and it is a chronic lifelong infection, which requires treatment daily. This is just akin to, for example, someone who's diabetic who needs insulin every day. People who have HIV need treatment every day. AIDS, or HIV, is amongst the top 10 leading causes of death in the world. It's number 6 over here in 2012. Just wanted to highlight that. And this is 2013 data, but currently there's about 37+ million people living with the virus. And you can kind of see where it's centered. A lot of it is in sub-Saharan Africa, as highlighted with this heat map of dark red. But as you can appreciate, no part in the world is actually free of the virus. So it is a global epidemic. And so this is the number of deaths that has gone on with HIV right from the time that we started first identifying it. And you can see there was a massive spike because of AIDS-related deaths. But at least we ended up buckling the trend in about 1996, because this is when we first started licensing antiretroviral therapy. Initially, it was just one drug. Now it's a combination of 3, which is called HART, which is highly active antiretroviral therapy. And since then, you can see that we've been able to control the number of deaths quite remarkably. And actually, to show you what that means is that a person who's HIV negative has approximately 60 years. For example, this is to say expected survival of a 20-year-old in a developing country who has access to, in a developed country, sorry, who has access to HIV treatment as easily as they can. So if someone's HIV negative, they have about 60 years to go. Someone who's HIV positive in the mid-90s had only about 8 years of life. And luckily, as of 2010, we've been able to add extra years of life, very close to someone who's HIV negative. So we're doing better with treatment. Of course, the treatment comes with its own side effects. But the highlight is that HIV doesn't have a cure yet, but we can treat it quite well if we give people access to medication. But that brings me to my next point. This is a map of, of people who have access to medication in the world. And what I really want to highlight is this is the percentage of people who have access to medication. And the bottom line over here is that people who need treatment every day do not have access to medication. You need this every day for your life, and so what we're finding out is that more than 50% of HIV-infected individuals in the world do not have access to medication. So what we really need is a cure. This is the clinical profile of untreated HIV infection, quite simplified. In the red, we've got the HIV viral load over here in the plasma, and in the blue line, we're following this CD4 T cell, which is the primary target of HIV infection. And as you can appreciate, as the viral load increases, there's an inverse correlation with the CD4 count. Although after initial set point, which is somewhere over here, it takes a period of years till the CD4 count gradually decreases, continually till a point that it gets to this cutoff point, which is 200 cells per microliter of blood, at which point someone is classified as having AIDS, which is the disease that HIV causes, at which point they'll succumb to opportunistic diseases and eventually die. What you can appreciate is for a certain number of years, the immune response will keep the virus in check, but will fail eventually. So it's not immediate death. It takes about 6 to 10 years for someone to kick into AIDS if they have HIV. One of the major obstacles to cure with HIV infection is that the virus establishes what's called a latent reservoir. Latent meaning hidden. And so the virus goes into hiding within hours of infection. Very early on, it seeds itself in lymphatic tissues or the target in cells that it wants to infect and just stays there. And HIV is a retrovirus, which means it integrates into the genome and keeps a copy of itself in the cells. So HAART can prevent, which is the antiretroviral therapy, can prevent replicating virus, but it cannot do anything with virus that has integrated already in the genome. This is what makes HAART lifelong, because if someone ever stops taking their therapy, the virus comes back very fast. And so we need to find a way to eradicate those few cells which have virus integrated inside of them. And what I mean by when I say HAART is lifelong is if you follow someone's viral load who's HIV infected, once they start their therapy, their viral load goes below this limit of detection. This is a clinical limit of detection. It's usually about 50 copies per ml. It doesn't mean that the virus ceases to exist after this, because with more sensitive assays, we can actually find it, but it doesn't show up with typical clinical assays. But if they ever stop taking their meds, the virus comes back within a period of 2 weeks, which is the evidence that even though you can't detect it clinically by the simplest assays, the virus is always there at some subclinical level. And that's enough for it to come back whenever it needs to. And this is what we mean by this hidden pool of HIV-infected cells that we need to target. And that's basically what comprises the latent reservoir. So we need to go after those cells. So I'm just going to quickly run over what latency means. I already told you HIV is a retrovirus. In a typical immune response, what happens is that you have these white cells or these CD4 T cells which get activated. They'll clonally expand, which is marked by these red cells. And then eventually, once the infection is cleared, many of those die and few of those become memory cells. This is the normal course of an immune response because you have memory cells to prevent future infections. If someone is HIV infected, when they undergo this course of infection, when these cells get activated, many of these cells will get infected with HIV because it prefers to infect activated cells anyway. And since the virus integrates into their genome, when these cells become memory cells as a course of their natural immune response, their genome will contain the virus and they will become a part of the latent reservoir. So what my project is basically is to kind of find this needle in the haystack, which is what a latent cell would probably look like. And what we want to do is to try to get the immune response to basically go in and find a way to kill, to make these cells visible to the immune system so that it can kill them once again. And what's important here is that there's an HIV protein called NEF, which actually plays a key role in hiding HIV-infected cells from the immune system. And what it does is it actually downregulates a surface protein called MHC class 1, which some of you might be familiar with. It's basically the main molecule that an infected cell uses to alert another T cell or another white cell, another immune cell that I'm infected, so please come kill me, take me out, because otherwise it's infected and it's going to pass on the virus. So just a quick primer on what NEF is. It's a very small protein, but the virus really seems to require it. In fact, it enriches for its copies in the first 8 hours of infection, so it seems to be very essential in the early phases and also to continue viral pathogenesis later on. As I mentioned, it plays a key role in immune evasion by downregulating this protein called MHC class 1, which alerts the immune system that something is wrong. In some clinical models, we've already known that if someone gets infected with a virus that is weak for HIV NEF, then they actually become better natural controllers of HIV. So there's already a clinical basis for this that we already are familiar with. And NEF you can always detect. Even despite therapy, you can find NEF protein to be detectable in the immune cells of people who are taking their medication regularly. So what NEF essentially does is this is an infected cell in yellow over here, and it needs to express this molecule class 1 and tell a green, basically a CD8 T cell, a killer cell, that I'm infected. Once these two interact, the CD8 T cell can kill the infected cell. This is done by MHC class 1 contact. But what HIV NEF does is it prevents this contact over here, which means that that cell is never eliminated. So NEF expression prevents HIV-infected cells from alerting the immune system that it's infected, thus the infection is persisting. So my project is to basically target NEF at a very early mechanism, at a very early step in its mechanism to downregulate this MHC class 1 protein, and to use these drugs to basically prevent all this class 1 downregulation and therefore allow the immune system to once again get the signal that it needs from these infected cells and therefore go out and kill them. And basically restore this handshake that we were talking about. So it's a very simple assay. What we do is we get latently infected cells. I'm not going to get into the details of the model, but we treat them with these inhibitors, and we isolate autologous CD8 T cells, which are the killer cells, the immune cells responsible for killing the HIV-infected cells, from the same donor. And we'll mix the two cells, and we'll basically ask how well the CD8 T cells or the killer cells were able to kill these infected cells, and how visible those cells were to these killer cells. And so I'll get straight into the results. In the first case, we're looking at this cytokine called interferon gamma. This is something that cytokines are responsible as a measure for CD8 T cells to go out and kill their targets. And so what we want to see here is we want to see more interferon gamma if the cells are able to see their targets better. This is killer cells co-cultured or mixed with uninfected controls, so that we don't see any particular improvement with these NEF inhibitors. But something interesting happens when we mix them with infected cells. So when we actually introduce these inhibitors, these 4 over here, to these infected cells, we can see that compared to our baseline negative control, we can actually see an increase in the response of the CD8 T cells or the killer cells by producing more cytokines. So we're actually making these cells more visible to the immune system. If we actually look by another measure, here we're looking at the ability, or we're looking at how many cells capable of producing virus are still left behind. Then we can see that if we introduce these inhibitors, there's less number of cells left behind compared to our control over here. And what that's basically telling you is that the killer cells were able to kill more of their targets, hence less cells capable of producing virus are left behind in the assay. This is by an ELISA, and we can also look at the same parameter by flow cytometry. And in the blue bars, we're looking at the percentage of cells that were eliminated compared to our baseline, so baseline's 100%. With each of these inhibitors, we're getting roughly a 50 or greater than 50% improvement in the amount of killing, as we can see with a reduction in the amount of cells that are left behind capable of producing virus. In the yellow bars, we did the same experiment with HIV that lacked the NEF protein. And so that's a good control, because if there's no NEF protein, these cells are already visible to the immune system, so the inhibitors don't seem to be able to enhance any sort of elimination of these cells. So one of the key findings that we have so far is that inhibiting this particular protein can enhance the killer cell response against these cells, and we can see that we're also making them more visible, and we're also able to increase their elimination, and the results are reproducible with a couple of different assays as well. And all this data, as I mentioned, is generated from patients who are actually HIV infected. This is not cell line work. We actually have blood donors who come into a clinic and give us their blood. So this is all primary cell work. And so with that, the conclusion so far is that we hope that this is going to be a potential strategy to develop a cure towards HIV infection by administering these drugs along with the therapy that we have to keep the virus in check. With that, I'd like to thank everyone at the lab. The guys in blue are the ones who are my students, who helped me a lot with this project, and of course my supervisor, Mary Ostrowski. I've got collaborators in Pittsburgh and my committee members as well, and of course the IMS. And lastly, but definitely not the least, to all the blood donors without whom none of this work would have been possible. So thank you.